Good afternoon, and welcome to the American Floral Endowment's Grow Pro webinar series. I'm your moderator, Michael Jasinga, and I am a grower for Bell Nursery in Springfield, Ohio. I help oversee and manage the production of a wide variety of ornamentals for hundreds of stores across the country. Today's session is on biostimulants for floriculture production. On behalf of the endowment, I'm excited to be a part of AFE's Grow Pro webinar series that features a new topic every month presented by an industry expert. The webinars are free to everyone thanks to the generous support of AFE sponsors. The views and opinions expressed in these webinars are those of the speakers and do not necessarily reflect the views or positions of AFE. As an independent nonprofit, AFE cannot endorse any specific product or opinion. Today's webinar is sponsored by Bloombeat and Oasis Grower Solutions. Bloombeat by Grower Talks is an online newsletter that provides timely tips, advice, and commentary for specialty cut flower growers. Oasis Grower Solutions, a division of Smithers Oasis, has been solving growers' problems with innovative substrate solutions and ancillary products for more than 55 years. If you'd like to learn more about our sponsors or are interested in becoming a sponsor for a topic, you can find that information on AFE's website at endowment.org/growpro. Today's session was pre-recorded in English by Dr. Michelle Jones. After the presentation, she will join us for a Q&A. Feel free to submit your questions through the Q&A feature or chat at any time. We'll answer as many as we can before the end of the hour. Unanswered questions may be answered through a separate email exchange. This session is being recorded and will be shared to AFE's YouTube account. Through YouTube's accessibility features, you can access closed captions in other languages. To get us started, I'd like to share a bit about today's expert speaker. Dr. Michelle Jones is a professor at Ohio State University in Horticulture and Crop Science Department and serves as the D.C. Kiplinger Endowed Floriculture Chair. Her current research and extension programs focus on the use of beneficial bacteria and biostimulants to increase nutrient use efficiency and improve disease and environmental stress tolerance in floriculture crops. She also works with greenhouse growers to identify and remediate ethylene damage in production greenhouses and improve shelf life of flowering ornamentals. Dr. Jones, welcome and thank you for presenting today on biostimulants in floriculture production.
Thank you for that introduction. I'd like to thank the American Floral Endowment for this opportunity and also welcome everyone to this webinar on biostimulants. I'd like to start this seminar with a little definition of biostimulants. So biostimulants are products that have really two main traits or characteristics. The first is that they enhance plant health, quality, or yield, and that's the trait I refer to as plant growth promotion. So this can't be due to the direct application of macro or micronutrients, but it has to be the result of an active ingredient in the product that makes nutrients more bioavailable, increases plant uptake. So it has to have an effect on the natural processes in the plant. The second characteristic is that biostimulants improve a plant's tolerance to abiotic stresses. These are environmental stresses like water deficit, even flooding, and high or low temperature stress. Biostimulants really have two main categories of active ingredients. They may have microbial ingredients and they may have non-microbial ingredients. And this is any various types of substances. Some of the most common substances or non-microbial ingredients are the humic substances, which are primarily humic acid and fulvic acid. A lot of products have protein hydrolysates. These are amino acids that are either derived from animal or plant sources. There are many different types of biopolymers. Chitosan is a popular ingredient in many biostimulants that you may have seen. There are also a variety of different types of botanical extracts from seaweed and algae and other types of plant materials. And then the second category is the microbial biostimulants that would include many different types of beneficial microorganisms, primarily bacteria or fungi. Bacteria primarily coming from the genus Bacillus, Pseudomonas, Rhizobium. Those are some popular ones. And fungi that may include endo or ectomycorrhizae, as well as trichoderma. So today I won't be able to cover details about all of these types of active ingredients. I'm going to focus primarily on microbial biostimulants that have beneficial microorganisms and talk a little bit about humic substances. So one of the biggest challenges with working with biostimulants for growers as well as researchers is the efficacy of these products is not very consistent. And I like to show this graph. It's a little bit kind of complicated. I'm going to walk you through it, but I like to show it because it illustrates some of the challenges that we first faced when we started studying biostimulants. And that is, we did a study where we went out to companies, we went to cultivate, we found 16 different products that were mainly microbial biostimulants. And we evaluated them in the greenhouse in soilless culture. And the purpose of this study was to see if we could use microbial-based biostimulants to enhance growth promotion at lower fertilizer levels, so with lower fertilizer inputs. So what we were really comparing here, and so I'll show you a little bit of the details here. We have the control plants, and this graphic just shows the number of flowers. So we're looking at growth promotion with the improvement in flowering, but also improvements in plant size, and other things were very similar. And when we grow petunias at what we consider kind of an optimal fertilizer rate of 150 parts per million nitrogen, we have really nice big plants, lots of different flowers, about 30 plus flowers per plant. And when we grow them at a lower fertilizer rate, which is this 50 parts per million nitrogen rate here represented by the black bar, no surprises here. There's fewer flowers, plants are smaller. So what we're really looking at is when you're looking at these 16 different biostimulant products, they were applied to plants that were fertilized at that low rate of fertilizer. So we're looking for that growth promotion, bumping the productivity or the flower number in this case up to what we see with that higher level of fertilizer. And what you can see here really is that there were only 2 products that showed any kind of growth promotion. And so what were these products? So in this experiment here, the blue bar is representing, this is the data from the product Cease, which actually is a biofungicide, but it has a microbial ingredient, bacterial ingredient, Bacillus subtilis, that does have growth promoting properties. The other product is a mycorrhizal-based product called Mycoply All-Purpose, and it has a number of different mycorrhizal as well as a few bacterial species. So one of the things that we started to do is look at what are the different factors that affect the efficacy of these biostimulants. And so there are many of these, and we'll talk about some of them today. One is fertilizer inputs, so kind of nutrient management is really important. Growing substrate components, the physical components of the substrate, as well as chemical properties of substrate like substrate pH. Growing in retail environments, so temperature, moisture, humidity, all of these things have an influence. Application methods and intervals, the host plant microbe specificity, especially with obviously the microbial-based biostimulants. And then any other inputs, biopesticides, plant growth regulators, any other inputs that go into that system, all can have an influence on the efficacy of biostimulants. One of the things that we've discovered over time when we've been doing this research is that fertilizer is really one of the most important production inputs that influences the efficacy of biostimulants. If you think about it in this way, if the plants are really receiving all the nutrients that they need, you may not see the benefits of biostimulants. There may not be anything biostimulants can do to really increase nutrient uptake. But higher rates of fertilizer, we tend to over-fertilize, especially with the phosphorus. These high levels of phosphorus can suppress colonization by mycorrhizal fungi, and they also reduce the activity of phosphate solubilizing bacteria. So this is important, and also I think it really illustrates the greatest potential benefit of the biostimulants is the idea of growing high quality crops with reduced fertilizer inputs. Here you'll see kind of a complicated figure with a lot of different pictures. So again, I'll walk you through this, but I like this photo because it really illustrates what I just mentioned with fertilizer levels and how important they are for biostimulant efficacy. And so what you see here on the top line of these pansies were untreated, so no biostimulant product. The ones in the middle were treated with cease. Again, that's the biostimulant that includes or biostimulant like product that includes Bacillus subtilis. So that's a bacterial strain, is QST713. And the bottom row of plants is the pansies treated with mycoplant endo. So again, that's the fungal based, mycorrhizal based product. And if you skip here to this middle column here, and at the bottom of the columns, you'll see that these plants are all receiving different rates of fertilizer. So it's a complete 15-5-15 fertilizer with different nitrogen based on nitrogen rates here. And when fertilizing the plants with 75 parts per million nitrogen, which we consider to be a kind of a low rate for pansy production, you can see that there's quite a big difference between the untreated plants and the plants that are treated with both cease and or the mycorrhizal based product. Plants are a lot larger, they have more flowers. There's definitely a growth promotion effects there. But when you move to plants that are treated with a higher rate of fertilizer at 150 parts per million, which we consider to be a kind of an optimal rate, you can see you've kind of lost the benefit of the biostimulants. You no longer really see a difference there or a growth promotion effect. So we tend to think that we're applying a rate of fertilizer that has been recommended and that's been maybe optimized for growth in our conditions. But even with water-soluble fertilizers, many of the nutrients that we apply are not always available for plant uptake. They quickly become complex with metals like calcium and iron. This is especially a problem with phosphorus that can bind calcium and iron and form insoluble complexes that are not available for plant uptake. These nutrients can also be bound tightly to charged particles within the substrate and also become unavailable. So the microorganisms that are in some of these biostimulant products can solubilize or mineralize these nutrients and make them available for plant use. And this is how microbial biostimulants increase the bioavailability of nutrients, and they can increase plant uptake. They basically make the plants use the nutrients that you apply more efficiently. And what that means to growers is that you can use certain biostimulants to produce high quality crops with lower fertilizer inputs. This picture illustrates, kind of further illustrates that effect. You see enhanced growth, health, and quality with less fertilizer. These are some zinnias. And if you look here on your far left, these are the zinnias produced with a higher rate of fertilizer compared to zinnias produced at the lower 50 parts per million rate of nitrogen. And you can see definitely the difference there. And when you compare the plants that have been fertilized, again, with that lower rate of fertilizer, but have had a biostimulant added, either mycorrhizae or a bacteria-based biostimulant, you can see there's quite a difference there in the growth, even comparable or better than plants that are receiving 3 times the fertilizer rate. So with some of these microbial biostimulants, you can get enhanced plant size, increased greenness, increased flower number, even when growing them with 1/3 of the rate of fertilizer. And so that growth promotion, you have bigger plants, you have more flowering. That is also accompanied by increased uptake of nutrients that contribute to that growth. And in the tissue nutrient concentrations, you can see increases in both macro and micronutrients. With phosphorus, we only saw an increase with the cease treatment, but in the other nutrients, there was a positive influence of both the mycorrhizal and the bacterial-based biostimulant on increasing nutrient uptake. One of the things that is become kind of a popular or something that many of the companies have been excited about and pursuing in the area of biostimulants is the idea of phosphorus solubilizing bacteria. And that's because phosphorus is one of the nutrients that can often become limiting and cause negative symptoms like the leaf purpling that you see here in this picture. And this is an example of some experiments we've done with a product called Laurus Vita, which is from Lallemand, and includes a phosphorus solubilizing bacteria, Bacillus velizensis. And in each of these pictures, you can see the control plants on the left, so no biostimulant treatment, compared to Laurus Vita treated plants. These plants, again, were grown with somewhat low fertilizer levels, but the primary difference here is that phosphorus was only supplied to these plants as an insoluble form of calcium phosphate. So you can see that the control plants really can't solubilize and take that phosphorus up. But when you add these phosphate solubilizing bacteria into the system as a drench into the substrate, then you improve the health and growth of those plants. One of the most interesting things too that we've come upon recently is really the effect of substrate pH on these biostimulant responses. And one of the other added benefits of some of these microbial biostimulants is the way that they're acting to increase nutrient uptake. They really can prevent your plants from suffering nutrient deficiencies if you have for example, a change in pH as you're growing. If the pH of your substrate increases to levels that would be a little high and where phosphorus, for example, might not be very available, these plants can protect from some of that deficiency symptoms. And you can see that here with the Laurus Vita. Again, this is the Bacillus velizensis that solubilizes phosphorus. If you're at a good pH, about 5.6, phosphorus is pretty available for uptake by these French marigolds. You can see that the plants with no biostimulant compared to the ones with biostimulant, they look pretty similar, maybe a little bit more purpling in the untreated plants at this pH of 5.6. But if you have the substrate, pH go a little bit high, this is pretty high, pH 6.8. You can see there in the untreated control plants that phosphorus is becoming unavailable. You see stunting, you see that purpling. These plants are not as great with the biostimulant as they would be at pH 5.6, but they look a lot better. They're able to take up more of the nutrients. So that's another thing that the biostimulants can do if you have some pH drift. Auxin production is another thing that has been highlighted a lot in the recent releases of different biostimulant products. This is a product called Azaroot that is produced by 3Bar and marketed, I believe, by Harrell's. And this is some research that they've done. I haven't worked with this product directly, but I've seen a number of different presentations from growers who've been using products like this that include auxin-producing bacteria. So auxin is a hormone that is involved in rooting, and growers often use a rooting compound that includes synthetic auxins to improve vegetative cutting, propagation, and rooting. And this shows you using a rooting compound versus treating with a product like Azaroot that includes auxin-producing bacteria, that you can get a little earlier root formation, a little more consistent rooting. So these are some interesting products that you might want to look for. I want to shift gears a little bit and talk about humic substances. So again, some of the non-microbial biostimulants. And humic substances are decomposition products of organic matter. They'll primarily be fulvic acids and humic acids. You'll see many different products with combinations of these humic substances. They increase water holding capacity of the soil and substrate. They improve structure, increase nutrient uptake, and stimulate microbial activity. So there's a lot of different modes of action by which these humic substances potentially can improve plant growth. We've worked with a number of different humic substance, basically containing biostimulant products. I just want to show a few examples of products that have worked pretty well in our hands. They've shown pretty consistent responses. This is a product called Micromate, which includes both fulvic and humic acids. And it can be applied either as a drench or a spray, or even just a one-time incorporation into the substrate when you're transplanting, for example. Some petunias treated with different rates of micromate. This is a drench application, and you can see the plants getting bigger. They have more flowers, up to the highest rate of 40 grams per liter of micromate. So when you look at the untreated plants compared to this highest rate, you have from 5 to 9 more flowers per plant with a micromate treatment. And interestingly, you also have plants that flower quite a bit earlier. So if you look at the highest rate, we've got about 7 days. So flowering about a week earlier, that's a pretty big deal. That can make a big difference in your production. So with the micromate product, the non-microbial biostimulant, the difference that we've seen with this compared to what I mentioned earlier with the microbial-based products is that even at the higher fertilizer rate, we see an increase or a growth promotion effect. These are plants that were treated with micromate, the higher rate, and the higher rate of fertilizer, considered the optimal rate, around 150 parts per million nitrogen. You can see that these plants still have more flowers. They're larger. It's not as many flowers as in the previous experiment that I showed you, which was conducted at the lower fertilizer rate of 50 parts per million, but you still do see some improvements. Not all biostimulants that contain humic substances will promote growth and flowering. So you do have to be careful, do some small scale trials. We've seen a lot of phytotoxicity from some of the products that we've evaluated. And this shows you a couple examples. One product that just included humic acid. You see the plants are stunted. There's some yellowing versus the micromate where you see the consistent growth promotion. One of the other benefits of biostimulants that include humic substances is that they provide a lot of sulfur. So you can use them if you have problems with sulfur deficiency, and you will see an additional benefit. We talked a lot about using biostimulants during production to improve plant growth and allow growers to reduce fertilizer inputs. Biostimulants can also be used to improve the post-harvest quality or post-production quality of plants. Many of the bacteria and fungi that are in these different biostimulant products were isolated from extreme environments, and they can therefore improve plant vigor or resilience under these extreme stresses. So plants don't usually experience extreme water or temperature stresses during production, but they can experience those during shipping and retailing. And so mycorrhizae and different plant growth promoting bacteria can protect plants from water deficit and high temperature stress. And these microbial biostimulants, therefore, are really providing protection during retailing and providing a higher plant quality for your customer. One of the things that when we first started this work and we were interested in water stress, looking at that from the post-harvest side of things, if plants dry down during retailing or during shipping, for example, we spent a lot of time trying to figure out if these different applications could kind of delay that wilting process or what really that looked like. And we didn't really get a lot of consistent responses. But what we did find was that when plants went through a severe water deficit stress or a drought stress, if they'd been treated with some of these biostimulants, that recovery phase was a lot quicker and the plants would remain marketable. And so really that's what's the most important, is that if your plants experience a stress and you're able, for example, with the water stress to rewater them and keep them marketable, then that is going to be a value from the biostimulant. And this can be seen here in an experiment with impatiens. plants on the left were treated with a biostimulant that included a species of Pseudomonas, beneficial bacteria, treated during production. When the plants got to flowering, water was withheld until they were extremely wilted, kind of starting to be a little on the crispy side, and then they're rewatered. And you can see 2 weeks after irrigation was resumed that the biostimulant treated plants have really resumed growth. They're larger, they're greener, they have more flowers, and they look a lot more marketable and aesthetically appealing than the plants that did not receive any biostimulant treatment. Similarly, this is an experiment with geraniums. It's looking at a couple different biostimulant products. These are the products that we've talked about. Laurus vita is the phosphate solubilizing bacteria. Micromate is a product that has humic and fulvic acid. And then cease is a bacterial-based product, Bacillus subtilis. And in this instance, geraniums were grown to flowering. They had already been treated during production with these products. They were allowed to dry down to very severe kind of wilting. Then they were rewatered. And this is what the plants look like a week after resuming the watering. Again, these plants were grown with a low rate of fertilizer to look at growth promotion initially, and then looking at water stress. And most striking here, you can see, is that cease provided a lot of protection from drought stress. These plants look better. They're much larger. They've resumed growth. They don't have the symptoms of the water stress and early senescence that you see in both the control and the products, the plants treated with the products Laurus vita and Micromate. In this experiment, we were looking at snapdragons, also interested in the impacts of biostimulants on vase life of the cut flowers. And in this experiment, again, we grew the plants at even in a more extremely low rate of fertilizer, only getting 25 parts per million nitrogen from a 15-5-15 fertilizer just one time per week. And then plants were treated with the different biostimulant products. You can see the extreme differences in growth. So that growth promotion with those products allowed them to utilize a very, very low applied rate of macro and micronutrients compared to the control plants. When all these plants did flower, we also found that the vase life of the cut flowers themselves was much improved, about twice as much as twice as long as the control snapdragon flowers. One of the other things to think about with regards to biostimulant efficacy is the method of application and the timing. One of the questions that I get a lot is why, if these microbial based products are colonizing the roots, do I need to reapply these products? So really, there's kind of two ways that these different products function. Some of them, like mycorrhizae, will colonize the roots. They form a symbiotic relationship with the plants. In this instance, they only need to be applied once, although it is suggested that if you pot plants up to a bigger size container, that you might reapply the mycorrhizae to improve that colonization as the roots grow out. But you also need to consider that this type of response may take some time. You apply a dormant spore of the mycorrhizae, it needs to germinate, it needs to find the plant roots, colonize, and this may take some time. So you need to treat the plants as early as you can, maybe treating in the transplant, treating in the plug stage instead of the transplant stage. And the second kind of mode of action by which these products work is they are microbial ingredients, for example, but they don't necessarily colonize the roots and provide a lasting effect. These microorganisms produce chemicals that act as signals. They may act as resources that promote plant growth. So these are the products that you will have to apply multiple times in order to see a benefit. Like pathogenic microorganisms, most beneficial microorganisms also have specific plant hosts. There are greater than 50,000 fungal species that form mycorrhizal associations with most of the terrestrial plants, 95% of plants. So this includes most greenhouse crops. But it doesn't include all greenhouse crops. So there are some important greenhouse crops like carnations and stalks that are non-mycorrhizal. Also, all of the Brassicaceae like kale, cauliflower, and broccoli. So if you do want to work with a microbial biostimulant that includes mycorrhizae, there's some good resources. For example, Mycorrhizal Applications has a nice database on their website. You can find out if the plants that you're interested in are mycorrhizal. Do they form associations with ecto or endomycorrhizae? When you are thinking about host range for the bacteria, it's a lot more complicated. So these rhizobacteria may have a broad host range. Some of them may have very specific hosts, but there's a lot less known about these different rhizobacteria than what is known about the host range of the mycorrhizal fungi. Just like any input in your production system, biostimulants have an added cost. So when do you consider using a biostimulant? So I like to tell people that if you think you'll be able to reduce other inputs like fertilizers or plant growth regulators, this might be a reason to try biostimulants to see if you can grow your crop, for example, at a lower fertilizer rate. If you can improve crop quality and yield, this includes with young plants. And the third reason is kind of the insurance side of things to protect from the unexpected. And that's where the stresses come in. If you want to protect your plants, for example, from water stress that they might experience during retailing or shipping. How do you choose a biostimulant product? There are a lot of different products out there, and they make very big and exciting claims. So you have to be as informed as you can about these products, and you have to know what is your goal for the product. Are you interested in growth promotion, reducing fertilizers? Are you interested in some protection of your plant from abiotic stresses? You want to choose products for which there are unbiased independent trials. See if you can see the data, ask to see pictures. You want to know if the product that you're interested in has been shown to work on the plants that you're growing. If you're growing in soilless substrates and the types of substrates under the conditions which you're growing, because many of these products have not been tested under greenhouse conditions with the types of plants that we're growing. Get very detailed recommendations for application timing rates and methods, because many of the products don't have a lot of detail that is provided with them. And you should always conduct in-house trialing to make sure that the product's working for you as you expect. So very briefly, I want to touch on a little bit of the ongoing AFE supported research that we're doing to kind of extend our research beyond evaluating some of the commercially available biostimulants. One of the things we're most interested in is the idea that biostimulants or microbial biostimulants can be used to improve nutrient use efficiency and also to protect plants from nutrient deficiencies and the symptoms associated with them, especially if they are in a situation where pH of the substrate might drift or it might not be optimal. So we focused on phosphorus deficiency, which you see or you may have seen on some plants that you've grown or seen others grow with the purpling, and then iron deficiency with the extreme leaf chlorosis. So we started this after we had done some of the experiments. We had a lot of inconsistency with the efficacy of the products we were testing, and we were concerned that it was because the microbial ingredients were really not interacting with our floriculture crops. They were not able to colonize and function or survive in, for example, soilless media that included peat. They really were not designed to be used in greenhouse systems. So we wanted to work to establish a bacterial collection from floriculture crops. So we worked with 15 greenhouses in Ohio to get 5 different plant species. We isolated bacteria from the rhizosphere, so the layer of the root that's just interfacing with the substrate. And we created a collection of over 1,000 individual bacterial isolates. And a lot of the research that we've been doing is on trying to find bacteria within that collection that have growth promoting traits of interest. We've focused on potassium and phosphorus solubilization, bacteria that produce iron siderophores so they can chelate and improve iron uptake efficiency, bacteria that produce ACC deaminase as a way of combating or mitigating stress responses, and then also bacteria that produce IAA, which is the oxidant involved in rooting. And then a large part of this is also then doing very detailed and relevant greenhouse studies to make sure that the bacteria actually work in production systems, in soilless media, in greenhouse crops. And just a few quick summaries of some of the things that we found. We found a lot of interesting phosphate solubilizing bacteria that can promote growth under the phosphorus limiting conditions or when pH might drift to above a range in which phosphorus is available for the plants. This shows you 2 bacteria that were used to treat French marigolds. These plants are grown with a decent level of nitrogen, potassium, and other ingredients, but no phosphorus from their water soluble, given again only the insoluble form of calcium phosphate. You can see that there's good growth promotion and they have less symptoms of purpling and phosphorus deficiency. So we've identified a number of really positive phosphate solubilizing bacteria that we think will have potential to move into commercial products. Similarly, we've been interested in iron. We grow a lot of iron inefficient plants in greenhouses that may exhibit severe chlorosis if pH gets too high and the iron becomes unavailable. These are bacteria from our collection that can chelate iron. So they're grown in a nutrient solution that doesn't include iron. Here, they're grown actually hydroponically in this experiment. And you can see when the bacteria are added to the system, and the only form of iron that we were giving these plants is a form of goethite, which is a mineral. So it's not chelated, it's not plant available. But you can see these bacteria can actually chelate and release this iron and make it available for plant uptake. So some very exciting work with iron. I'm very grateful to American Floral Endowment for funding these types of projects. So we want to wrap up with just a little bit of a summary. Biostimulants can improve plant growth, health, and stress tolerance, but getting consistent responses is definitely a challenge. The efficacy of these products is influenced by many different factors that we talked about today. And one thing to consider, you may notice that it looks like your biostimulant products may actually have the same active ingredient between many different products. You may have the same species of bacteria, for example. All products are not created equally, though. The formulation of that product is really extremely important to make sure that it's providing the nutrients that that strain of bacteria needs to get going and to function once you've added it to the plant. So it is really important that products have been fully tested. And lastly, if your goal is to reduce fertilizer use, trial the biostimulants starting at about 75% of your normal rate to see if that works, and you may be able to reduce it even more after that. So I want to finish by thanking American Floral Endowment, all our other funders and researchers in my laboratory and other collaborators. Very grateful for your support. And there is a lot of this information is also available on eGrow resources. And with that, I would like to entertain any questions.
Dr. Jones, thank you so much for your presentation. As a greenhouse grower myself, I found it really informative. Before we get into the Q&A, I just want to make a quick reminder for next month. We will be hosting another webinar with Dr. Johanna Del Castillo. The presentation will be on root rot and managing omycete pathogens in floriculture production. It'll be on Tuesday, January 20th at 1 p.m. Um, and you can go ahead and register now at endowment.org/growpro. And I'm going to go ahead and open it up for Q&A. So I have some questions here in the chat in the Q&A box, but feel free to continue adding questions as we go through some answers here. The first one I have is from Karen. In your experiments, how often are you applying the microbial products?
Okay, that's a great question. Can you hear me okay? Yes. Okay. Thank you. Um, the different microbial products, if they're biostimulants that we're using from companies, we're following their recommendations based on label. Many of those are applied multiple times in the experiment, sometimes weekly, sometimes biweekly, based on label. When we're doing our experiments with our kind of microbe discovery kind of pipeline, we've been applying our bacteria every week as a weekly application, just to make sure we're getting them there in the system. Now we're doing follow-up experiments in order to isolate them at the end of the experiment and determine if they're there. But the answer to that question is that it really, really depends on the products. Some of them we're incorporating at the beginning of the experiment one time only, like the mycorrhizal based products, the micromate, some of the humic substances. They work really well if you incorporate them in, and that's it. You don't have to apply those again.
Awesome. Got another question here from Jan. So you talk about about stress, about drought and heat stress. Are there any products that you could think of that address cold periods as well or cold stress?
Cold stress. I've seen that, and I've seen the literature more on the scientific side that has shown a few bacteria species, for example, isolated from extreme environments that can provide some stress protection to plants. Unfortunately, I don't know of any commercial biostimulant products right now that are specifically indicating and showing a really great response as regards to cold stress. But that would be definitely very interesting, for early spring bedding plants getting outside and also into the fall. So I'll definitely look into that and try to look at evaluating some of those types of products if they're out there.
Another question here from Ben. In your experience, is there a minimum spore count, looking at CFUs per gram or CFUs per milliliter that we should look for when choosing a biostimulant? I see products with less than 50 spores per gram for numerous products. Thanks.
Yeah, yeah. So usually you're going to be looking at CFUs that are in the millions and billions, like 10 to the sixth, 10 to the seventh. Um, and so again, it's really hard to put a number on all of those, just because it's very different based on what the microbial, what it is and what it needs. And one of the challenges is some of these products are kind of a consortia. They're like a soup of a lot of different organisms. And so it's really not clear what the final CFU will be for that product because it's not always known that those microbes will really interact in a positive and synergistic way. So those are some of the challenges of products. So I would look more than looking like that. I would look at companies that you have worked with in the past that have products that work maybe on the biopesticide or other, and just get some good information from them about the product and um, how it's worked and what kind of plants and systems it's worked in, and then what are their recommendations for applications. I think that's more important really than going out there with the shopping and trying to see the difference between, because they're not always active and alive, even if they say there's like a billion CFU or 10 to the eighth or 10 to the ninth. It's a nice selling point, but it isn't always really super accurate.
All right. Got another question here from Sarah. So, hi. In your studies, did you have other products containing Bacillus subtilis QST713? We have other products, fungicides like Serenade and Rhapsody, containing the
Mhm.
same Bacillus subtilis strain. Should it give the same effect if it's the same bacteria?
So we have used Serenade as well as cease, and it is the same active, and we have seen similar results with that. They're the same. I think they're pretty much the same product marketed to different kind of crops and markets. That being said, sometimes if it's got a strain name, it should be the same. If it's QST713, it should be the same. A lot of times you won't see strain names indicated. You'll just see a species and you'll have Bacillus subtilis, and that won't necessarily be the same as the QST713.
Awesome.
Yeah.
I have another question here. Have you had the opportunity to test any nitrogen-fixing bacteria during your trials? If so, what were your results? And they also said, thank you for your presentation. All this information was.
Thank you. Thank you. We haven't really focused on nitrogen fixing bacteria from our standpoint. We've thought about doing a little bit of field work with some of our colleagues here on nitrogen fixing. So we have evaluated a number of different products that have consortia of bacteria that will definitely have nitrogen fixing bacteria in them that have had some positive but not always very consistent responses, and it's nothing that I can kind of pinpoint that it's the nitrogen fixing bacteria. We generally think that when we're applying our fertilizers in the greenhouse, nitrogen has not always been much of a problem for us. That's why we focus on the phosphorus and iron and things that can kind of get out of whack really quick if you have pH issues and if you have um, a lot of the leaching, phosphorus, you can lose a lot of that through leaching. So unfortunately, I don't have a lot of good specific recommendations for you about the nitrogen fixing bacteria.
Alrighty. Got a question from Katie. Are there crops that you would not want to use biostimulants for drought response effect? Well, yes. For drought response effect while using a lower fertigation rate. For example, impatiens are typically grown on the dry side with reduced fertility, and the potential for activating the drought response from the biostimulant would cause even fewer irrigation cycles and as a result could run into increased nutrient deficiencies.
That's a complicated question. Let me think about that. Okay. So um you asked, oh, you moved, oh, it moved, so I can't see it anymore. um so the concern is kind of to over activate their response to drought, right? So that you my answer kind of generally to that question is no, I don't really think that's a concern. One of the products we've studied a lot of products with regards to drought stress and kind of that retail side that focus on abscisic acid. So what they're doing is really shutting down the stomates and preventing loss of water loss through transpiration. And as you know, the transpiration stream cools the plants, and that can cause some heat damage. So those products are useful only to a certain extent because the different bacterial products, for example, the microbials, they're improving water uptake. They're not necessarily reducing loss by closing stomates and things that would cause kind of that negative impact. So I don't think that you would run into a problem with that. Um, if that really answered your question. I could think I kind of lost track of the details of your question.
Yeah, and Katie, if you have any follow-ups, feel free to pop them back in the chat and we'll readdress them as well. All right. Question from Laura. Did you check your roots for mycorrhizal colonization during your studies?
In some of the studies, we have done some follow-ups with mycorrhizal colonization checks, either in-house or we've worked with some of the companies where they've confirmed a colonization. So the mycorrhizal based products, we've had a lot of luck with them. but not always consistently. Like if we change products and we change batches and lot numbers, we've done entire experiments where we haven't had a response and we have confirmed at the end that there was no colonization. So um, the products in general with mycorrhizae, I like to think of them as kind of a gateway to biostimulants. They're kind of the easiest ones to start with. But again, the products are really different. So it is a really good idea. Even with the companies, you might be able to get them to do a check of your roots at the very end if you're really going to be interested in using their product. Because the mycorrhizae, you can actually do some staining and check if you have the ability to do that. Some of the other products, the other microbes, the bacteria, we really can't do a lot of checking for the commercial products at the end.
This is a nice follow-up question from Riley. Do you know if there's any negative interaction or competition between trichoderma and mycorrhizae fungi?
I don't know. I haven't worked a lot with trichoderma. I don't know of any negative interactions with those products. The trichoderma based products, they tend to, like Root Shield is a really great example of a trichoderma based product. They tend to be just the single trichoderma, maybe multiple species, and that may be for a reason. Maybe they don't play well with the mycorrhizae. Um, I tend to like products that don't try to mix a bunch of different species of fungi and bacteria because unless the company has really done a lot of R&D, a lot of research before it's released, they really cannot possibly know that under the situations in which you're going to apply that all those things are going to act synergistically. So I think that sometimes it's better to start with products that have single or similar active ingredients to kind of avoid those issues.
And then I have an anonymous question here. In the same line of mycorrhizal colonization in the roots, how do you determine the best colonization result? They give some examples like UFC or physiological response related to the soil health.
Um, okay, so I'm not sure like if this question related to like when you apply a product, do you see that particular organism, or is this in general the question about native, like within us, for example, if I have a container of a peat-based substrate, what is the native microorganism or microbiome population in that container versus hydroponics? Like, is that the question? Um, yeah, okay.
Yeah, whoever
S
wants that, if you don't mind going
so
ahead and
whoever
putting
said
a
that, I
little
don't.
follow-up info in there, we'll be happy to try
Yeah,
to address it before the
yeah.
end of the call here.
Well, I can just tell you in general that, for example, if you're looking at native microbiomes, native kind of populations of these biostimulant, perhaps containing bacteria, populations are much more diverse in soil. And as you move into soilless substrates, um, did I say much less? Much more diverse in soil. And as you move to soilless substrates, which include peat or core, um, much less diversity. So fewer numbers in general, much fewer numbers of different species. And then when you move into a liquid hydroponics, it's even less. But there are still microorganisms working in all these systems that come from the substrate, that come from fertilizer, they come from your water. Um, and they all have the potential to be beneficial. A lot of times we're focusing on them. If they're pathogens, we're really worried about it. We're not usually thinking about all the good things that we might be bringing into the system.
All right. Question here from I'm sorry, Ken Coles. Have you tested combination of products with mycorrhizae with mycorrhizal helper bacteria such as Pseudomonas or Bacillus strains, and combining it with humic and fulvic acids and others, um, and basically they just list a bunch of different combinations. So have you tried combining different biostims together and seen any results?
We've worked with a couple products that already have kind of I mentioned one of the micro apply um, products that we used initially had bacteria and the mycorrhizal fungi. That product is nicely formulated. It worked really well with that combination. We've taken a few of the biostimulants that have worked well for us with the fungi and or the bacteria and combined them with the humic substances. You don't necessarily get like an additive effect of better growth promotion. I think the benefit there is you're creating something as long as you don't have any negative interactions there. You're creating something that has multiple modes of action by which it can promote growth, improve stress tolerance. You're kind of priming the system to be ready to be able to respond to more. So we have seen some benefits of having, for example, the humic substances with the bacteria based products. The humic substances actually improve the quality of the soilless substrate. They make that environment more suitable for the bacteria. So the bacteria grow better. They do more as far as growth promotion. So there are a lot of good combinations. I can't really list too many like specific ones for you right now, but there are definitely some benefits to the different active ingredients kind of thing with the biostimulants.
I got a question from George. What do you think about algae extracts?
Algae extracts. Um, so we've read quite a few papers recently, on the scientific literature side of thing with algae extracts, having some positive growth promotion. I haven't done anything personally with algae. We've got a couple products kind of in the queue with algae. Some companies we're talking with. Also, chitosan is one that I've seen some positive results from colleagues that have done experiments with them. So I don't really have an algae-based product that I could recommend or that we've actually, really worked with specifically. But I do see in the scientific literature that there's some good basis for those products working well. Again, it's going to be formulation of the actual product and how well that will really work.
Got time for a couple more questions, and then we're gonna have to wrap up. So another question from Tom is metagenomic analysis a good way to understand diversity and function of microbial communities?
Is meta is Did you say is metagenomic analysis?
Yeah.
Yeah, of microbial communities. Yeah. So that is one of the things that we're doing, different approaches to looking at the different species of bacteria, for example, that might be in the microbiome, understanding so metagenomics and other microbiome types of approaches. There's multiple different approaches to determining what your native populations are in different types of substrates. So that's one thing that we've been really interested in. It's really kind of difficult because it's very, it's influenced by a lot of different things. Where did the peat come from? What was the source? What was the processing? So even with the peat substrate, everything's very different. But one of the other things that we've been interested in using metagenomics for and understanding kind of the microbial communities and how they're influenced by the inputs that we're putting in there, right? So, regardless of what they are when they started, if you had a plant growth regulator like Ethifon with your very acidic pH, what are you doing to the beneficial microbial population that's already in your container? And with our fertilizer applications with different pHs. So are there conditions that we can say are the best for promoting what's already beneficial in our system? Maybe we don't even need to add them as biostimulants.
Awesome. Unfortunately, we're going to have to go ahead and wrap up. There's a lot of thank yous that are flying through the
Yeah.
chat and the Q&A, and I'd like to thank you as well. It was very informative for everyone. I know we didn't get to answer everyone's questions, but feel free to reach out to Dr. Michelle Jones. I think
Yes.
her information should be on the AFE website, and it sounds like she'll be happy to answer any other questions that we have. Be sure to mark your calendars again. Just want to remind everyone for next month's webinar. And you could always check out past webinar recordings and grower-related resources, research reports, all available to free thanks to industry support at endowment.org/growpro. And if you found these webinars valuable, we want to encourage you to make a donation to AFE to support future resources like these. If you make a contribution by December 31st, your donation can qualify for a special gift match, which will triple your impact. The link will be in the chat if you'd like to make a contribution. And then we also would like to ask that you please complete a brief survey about today's session, where you can suggest additional topics and help us continue to improve these webinars going forward. Once again, want to thank everyone for joining us and wish everyone a great day. Thank you.
Thank you.